Okay, good morning, good afternoon, and good evening, wherever you are joining from. So today, we're going to have this seminar on nitrogen accumulation, recycling, and uptake, a framework for optimizing nutrient use efficiency and BNI efficacy in agroecosystems. My name is Tek Sapkota. I lead the climate change science unit here in CIMMYT. Today's speaker is Professor Stuart Grandy. I will briefly introduce him, who he is, and what he is working on. So Professor Stuart Grandy is the Professor of Soil Fertility and Biogeochemistry at University of New Hampshire. And he's also co-director, founder and co-director of the Center of Soil Biogeochemistry and Microbial Ecology at that university. He earned PhD from Michigan State University and postdoctoral fellowship at University of Colorado. At University of New Hampshire, he leads interdisciplinary team that examine how agroecosystem management influences soil health as well as the microbial processes driving soil organic matter formation and nutrient cycling. He has published more than 130 papers in high-impact journals, and he is one of the highly cited scientists of Web of Science in 2022, 23, and 24 consecutively. And he's fellow of the Soil Science Society of America and the winner of Soil Ecology Society Professional Achievement Award. So his research applies the novel microbial explicit model on the fundamental understanding of soil biogeochemical processes to improve the agroecosystem processes and management in collaboration with academic, industry, and research stakeholders. With this, I will invite Professor Stuart Grandy to present on this topic. Thank you. Over to you, Stuart.
Thank you, Tek. It's great to be here. I'm really excited to be at CIMMYT and appreciate the wonderful hospitality, Tek, for having me here and all the others that I've met. And it's a really exciting time for me to be in this place that has such a rich history of impactful agricultural research. So thanks to all of you for hosting me and having me here for this talk. So as Tek said, I'm a professor at the University of New Hampshire. I did my PhD at Michigan State, a postdoc at the University of Colorado. So today I'm going to talk about some of our work on nitrogen and sort of its evolution and how we're thinking about nitrogen. And then I'm going to end my talk and transition briefly into the integration between some of my historical work and the work that we're doing on the BNI, the biological nitrification inhibition in collaboration with CIMMYT. And so to start out, my work early on was on pathways of soil organic matter formation. My postdoc with Jason Neff at the University of Colorado was really challenging some of the historical ideas about how organic matter forms and really testing some of the entrenched ideas about humification and the role of aromatics and plant-derived materials in soil organic matter formation. And through that work, we began to see a lot of evidence that much of the organic matter in soils, especially that organic matter that's associated with minerals, or the more stable fraction of organic matter, was not derived directly from plants, but was first processed by microbes. And that has real implications for how we think about soil organic matter management. And since that time, so that was around 2007, we've continued that work, and I've had various people come in and work on that, including Emily Whalen, who we recently published a paper with revisiting the historical arguments and evidence for different pathways of soil organic matter formation. So before I talk very briefly about this and then transition from that into nitrogen, because I want to provide that background, how we ended up in this place where we're thinking a lot about nitrogen, I think it's helpful for you to have a bit of background. And so, especially if you're not a soil scientist, I don't want you to be lost in terms of jargon that us soil scientists routinely use. And so, one of the things that soil scientists do to better understand soil organic matter is we separate it out into different fractions. And there's a number of ways to do that. You can use a density fractionation, which is a simple flotation in a high density liquid, and that gives you what we call a heavy fraction or a light fraction of the soil organic matter. The stuff that floats, it sort of looks like tobacco. It's younger, it's more plant derived. The stuff that sinks is associated with the minerals, and it has more microbial chemical signatures. It's a little more stable because it's associated with the minerals. And another way that we fractionate is just with a sieve, a 53 micron sieve, which is the separation between sand and the silt and clay fractions. And we simply disperse the soil, put it through a 53 micron sieve, and what remains on that sieve is what we call light fraction organic, or it's called particulate organic matter. It's similar to the light fraction. It is plant derived. It looks a little bit like tobacco. It's younger. And what goes through that sieve is more mineral associated organic matter. And we call it MAOM, mineral associated organic matter. That's what goes through that 53 micron sieve that excludes the sand. And so you'll hear me today refer to MAOM a number of times. So you can see these are the different properties of particulate organic matter in MAOM. The POM has a higher C to N ratio. It's more plant derived. The mineral associated organic matter consists of smaller nitrogen rich molecules, a lot of proteins, amino sugars. It's often associated with nitrogen mineralization. You can see over there an approximate distribution of organic matter among the mineral associated and particulate organic matter pools. So we first became interested in this pool of organic matter from a carbon perspective and thinking about how does organic matter form, again, putting it into the context of these questions around is humification a real process? Is this the correct way to think about how organic matter forms? And this pool, the MAOM, becomes very important in thinking about that because it is so much of the soil organic matter. And we know from using isotopes that it has a longer turnover time. It's more stable. And so as we think about building organic matter in soils, we want it to be in a form that's somewhat more stable. And so there's been a lot of focus on where does this organic matter derive from? And we thus do this sort of separation to explore those mechanisms of organic matter formation. And so in 2007, I published this paper with my postdoc advisor, where we went up to the San Juan Mountains. You know, I'd always worked in agricultural systems, so this was quite fun and novel for me to be in very, very high mountains and collecting soils, and it was all very, very exotic. But we brought them back, and I separated out these soils into a light fraction and heavy fraction. Again, you can think about the light fraction as that particulate organic matter, the heavy fraction is MAOM. And we did a chemical analysis using a spectrometric technique called pyrolysis GCMS. And we were able to look at the broad chemical characteristics of these different fractions. And we noticed that the heavy fractions were filled with nitrogen and microbial lipids and carbohydrates. So this really was quite eye-opening. So this fraction of organic matter that makes up the majority of organic matter in soils and that is the most stable was appearing to be mostly made up of microbial dead bodies, not plant derivatives like lignin. Yes, there was lignin in the soil, but it was all in this light fraction. And this was raising a lot of questions in our mind about these ideas that most organic matter in soils is derived from what microbes can't break down, the lignin and other complex organic molecules that are left behind by decomposition. And this was suggesting a very different model whereby microbes break down everything because that's what they've evolved to do eventually. And it's their bodies that associate closely with the mineral surfaces and form stable soil organic matter. And I'm not going to go into great detail in this, but we did a number of projects in this area for a number of years. And I think it really kind of came together with the work of Cynthia Kallenbach, who did just a very simple benchtop experiment where she fed microbes glucose in artificial soils. So she took pure minerals, clay-sized particles, and inoculated them with microbes, gave those microbes glucose, and after 16 months of incubation and feeding microbes glucose, without any other kind of plant inputs, she ended up with organic matter that looked chemically identical to the field soil from which the microbes came from. And again, this was not evidence from the field that all that organic matter is derived from microbial cells, but it was sure very compelling evidence that the microbes can produce organic matter with the chemical complexity that we see in field soils. And she built on this and started to really open the door to thinking about microbial traits and organic matter formation and things like carbon use efficiency of the microbes and turnover rates. And then we followed that up with work by Emily Whalen, this other student who really built on the trait framework, but I'm not going to really go too in depth into her work today. So with time, we found this important role of microbes. Other people in the soil biogeochemistry community were also working on this and finding similar kinds of very strong evidence for the role of microbes in soil organic matter formation. And we developed, together as a community, a new way of thinking about organic matter formation. And part of this, as we put microbes at the center of it, was really realizing the importance of roots in soil organic matter formation. And I know, for example, the plant breeders, there is an area of plant breeding that's very interested in, how do we get more roots into the soil? And this is why. This is an experiment we did where we labeled the growing plants in the field with 13C CO2. So this was an enriched but non-radioactive isotope, but it allows us to track all those plant materials through the soil. And this is the amount of plant material in the soil after a couple years that was root or microbial or root or shoot derived. And you can see 2.6 times more cover crop root carbon than shoot carbon was present in the soil after about 18 months after this cover crop was killed. And we also built on this with other work, for example, by Andrea Yilling, where she showed, and others have also shown this kind of dynamic where tillage and no-till is great for building up POM in the soil, but to build up the MAOM, or that mineral associated fraction, you really need to get more roots into the system, and that's where the cover crops become quite important. And so for a number of years, we were really focused on organic matter dynamics. We did a lot of work on crop rotations and thinking about the role of the different crop functional groups and diversity per se in organic matter formation, and trying to think about our organic matter work in more of an applied context, in the context of cover crops. Okay. So somewhere along the way, probably when I was on sabbatical, I started to think, well, I'd like to, be looking to do some other things over the next 5 to 10 years. I've worked on these questions of microbial organic matter formation for a long time, and they've been fun. But I didn't want to just continue to do iterative work in that area and, do ever more sort of reductionist experiments. And I got to thinking about this question that I'd always had about, where and how do plants get nitrogen? This very broad question. And it seemed to me like there was a lot of a surprising amount of uncertainty in this area. And, just anecdotally, I've been on countless farms where farmers speak to their ability to produce very, very productive crops, achieve the kind of yields they're looking for, using alternative kinds of nitrogen management. And that always was very intriguing to me. What does that nitrogen balance look like? How would you pull off a more synchronous nitrogen cycle? And I also thought a lot about my time working in the Midwest, where we had these beautiful prairies that were extremely productive, but incredibly tight with respect to the nitrogen cycle. You would see almost no nitrate in these soils, which interestingly, I'm now going back and wondering in my mind if we haven't overlooked biological nitrification inhibition is the reason for that, because I always heard other reasons, and I might have to go and revisit that in a grant soon. Anyway, I became intrigued by the fact that these were very productive systems that were losing very little nitrogen to the environment. And where were they generating all of this nitrogen from? And when I would talk to other, soil people and biogeochemists, they would, talk about, oh, it's the turnover of that light fraction, that tobacco type of material, or, the turnover of the microbial biomass that was supplying the nitrogen. But this just didn't really add up. Like from a mass balance perspective, it didn't really make sense. And so along came Andrea Yilling, and she really began to probe this mineral associated organic matter pool that we'd historically thought was really stable. And the reason we think it's stable is because if you carbon date it using C14, it can have an average age of hundreds to thousands of years. But remember, it's a huge amount of the organic matter in soils, like 80%. And there are thousands and thousands of kilograms of nitrogen in a reasonably productive topsoil. So if only a small percentage of that, 5 to 8%, is turning over rapidly, that's a huge hundreds of kilograms of N per year that's potentially cycling through and out of and into that MEAM fraction. So we began to think about that. And so Andrea came along and developed this conceptual model where she argued that yes, MEAM can be stable, but there's a wide variety of mechanisms by which organic matter associates with mineral surfaces. Some of them are very stable. There is some organic matter that is covalently bonded to a mineral, but there's a lot of it that's much more loosely associated with the mineral surface. So it's held under some loose sort of attraction to it. It's probably less mobile in the soil, like some of the soluble molecules, for example, may not be just freely moving through the soil because of their association with these mineral surfaces, but they're still not tightly bound. And there's a lot of different kinds of chemical interactions that are represented in the soil. And at the same time that we were, as a community, making a lot of progress on understanding really the geochemistry of that MEAM fraction, there was a huge amount of work happening on the plant rhizosphere, that area under the influence of the roots. And it's where the plants and the microbes are really tightly interacting. There's a ton of signaling happening. And it's also where there's tons of nutrient turnover and cycling as microbes release nutrients and take them up and release them and take them up. And the more we explored some of the literature that was emerging with regards to understanding the rhizosphere, it was becoming quite clear that there were mechanisms by which plants and microbes can mobilize this mayam nitrogen. And Andrea, in her 2018 paper, which is again represented on the right here, laid out all really two different very concrete pathways by which plant microbe interactions could serve to liberate organic nitrogen associated with the mayam N. And so this became very exciting because we're no longer talking about needing huge pools of soluble N, and we're talking about the possibility that we can have this continually cycling fast turnover mayam N that can become available at the times that the plants need it through microbe plant interactions. And so this is sort of what we've been building on over the last few years. And Andrea just published this very lovely update to some of the ideas we've been developing that Amanda Daly, who's a research assistant of mine at UNH and responsible for a lot of our field work, she's been very much a part of it and really dives in this work into what are the processes by which mayam nitrogen can become mobilized through environmental factors and plant root microbe interactions and management like tillage and potential for this to have a real impact on ecosystems, both natural ecosystems and agricultural ecosystems. So we've been building off of this work with a lot of experiments. And so one thing that a recent student of mine did in collaboration with industry, because one of the exciting things that we found is that, when we first started putting these ideas out, some of the biogeochemistry community thought, yeah, this is cool. We're seeing evidence of this kind of thing. But industry was not interested. But now we're finding that a lot of industries are very, very interested in this because they want to improve nitrogen use efficiency, and their approaches just haven't been working. And so they also see evidence in their own on-farm work and collaboration with farmers that the model that we put forward in terms of this cycling through microbes and mayam and remineralization seems consistent with some of the timing of nitrogen availability that they see. So the first thing we did was, well, let's test this idea that like even a large slug of fertilizer can go through microbial biomass and enter the mayam pool. So this student, Manjot, is still working on this, but she basically applied nitrogen at different rates, going from left to right here, very high rates over on the right. I don't remember exactly, maybe 150 to 200, the equivalent of 150 to 200 kilograms of N, very, very high, like parts per million. And this is the amount of inorganic N that ended up in the microbial biomass and mayam after only 14 days. So this new inorganic fertilizer input, you can see that it was quite common for anywhere from 15 to more than 30% of this nitrogen at a single time point to have entered into the organic nitrogen cycle. And I think it's important to keep in mind that microbial cycling is continuous and iterative. This is a single time point. So you can speculate how much of the actual nitrogen that's been applied, the total nitrogen that's been applied, at some point went through a microbe and became associated with that mineral surface. It's no doubt much higher than this number, which is a single point 14 days after application. And so then we also were asking, okay, what about in the with other kinds of inputs, again, we're using N15 for all of these experiments. This is some work that a student of mine, Emma Berg, has been working on, and she used a low organic matter cropland and a high organic matter grassland, and looked at the incorporation of 15N fertilizer into the MAOM pool. So in the case of the root litter and fertilizer, root litter, shoot litter and fertilizer, and shoot litter, in all of those, it's the litter itself that's labeled. And so right away, you see a very high proportion of, well, let's just start with the fertilizer. So the far left, you can see in the grassland that has higher organic matter, 20% of the fertilizer is within 3 weeks is ending up in MAOM, probably because it's being immobilized by the more active microbial community and more organic matter, and about 13% in the very low organic matter cropland. When you have root litter and fertilizer, again, you can see that there's very rapid incorporation of that nitrogen into MAOM. And so what this is suggesting is that fertilizer inputs and litter inputs are very quickly entering that MAOM nitrogen fraction. Now, we can't have a dynamic where nitrogen's just continuously going in, right? It needs to be going in and coming out and going in and coming out, because presumably most of these systems that are at some kind of equilibrium. And again, let's step back and ask, why does this matter? It matters because it's taking this nitrogen out of a form that's readily lost and putting it into a form that's a short-term reserve of nitrogen, and one that can be remobilized by those plant microbe interactions. And again, it appears that lots of N, both originating from fertilizer and shoot and root litter, does cycle very fast through this MAOM fraction. Both this study and the previous one suggest strongly that having some organic matter in the soil really helps move that inorganic N into the microbial biomass and MAOM, so it's not just sitting there. So we've been very interested in then how accessible is this newly formed MAOM. And what this shows is, let's see, this is hot off the press data. The total amount of MAOM formed are the orange and purple bars. And the purple bars represent how much of this nitrogen was easily extractable by chemical means. So we were trying to use a proxy for a plant's ability to access this MAOM. And so now we're beginning to get into some experiments where we're actually using N15 labeled MAOM to be able to track the nitrogen that's entering the plant and directly measure plant's ability to uptake N15 labeled MAOM. But this is sort of a nice proxy. And again, it shows like somewhere around 15 to 20% of the new inputs are extractable nitrogen, and maybe about half of the total N that forms is extractable. So again, we're getting rapid formation of MAOM, and some of it's entering a longer term pool, some of it's entering a pool that turns over much faster. Okay, so I don't want to go into the details here, but this did lead us in a recent paper to just sort of re-emphasize the role of organic matter, nitrogen accumulation in nitrogen use efficiency. And people have talked about nitrogen use efficiency for a very long time. And so, I'm not putting this forward in a way like no one has ever thought about this before. In fact, you can find papers where they refer to this term as, at least there's one paper is, I think, ecosystem nitrogen use efficiency. But here we wanted to emphasize that from an environmental standpoint and a long-term fertility perspective, any nitrogen input that enters that fertilizer pool or the organic, or that enters the plant pool or the organic matter pool is a win, and we should be considering both in nitrogen use efficiency. We tend to think of NUE as how much of the N that goes in is taken up by the crop. Of course, plant biologists and physiologists can have lots of other ways to think about NUE in terms of how much is going into the grain and into different parts of the plant. But this is something we called NUE agroecosystem because we really see the value in the intermediate to long term of also building up that organic N pool over time, and that this is a really important way to manage our nitrogen over different timescales to improve NUE. And so we have been, really diving into these different phases of the nitrogen cycle and really going back and revisiting just the basic principles of ecosystem science and thinking about nitrogen in agricultural systems from the standpoint of accumulation, so how it gets into the soil, and nitrogen cycling and retention, that ability for microbes to immobilize that nitrogen and then remobilize it. So that's cycling in and out of the mineral associated pool, often motivated by the activities of plants. And then also really thinking about the uptake of nitrogen. How do plants forage in the soil? What's the role of mycorrhizae? What's happening along that root plane in terms of enzyme production and communication with the microbes and subsequent mobilization of the nitrogen in terms of uptake, and that to better improve NUE and improve the synchrony of our agricultural systems, we found this to be a really nice way to sort of partition the nitrogen cycle into different sort of discrete, really critical steps in nitrogen use efficiency, and each one, while interacting with each other, provides a different set of approaches and experiments that we can be using to approach this question of NUE. And as you'll see later, it will circle around when we talk about the biological nitrification inhibition. This model also provides sort of a way to think about agricultural systems and what the priority should be of a system based on its organic matter, based on its vulnerability to nitrogen loss and its inherent fertility. And so you can think about systems where, really you just have to get nitrogen into the system, low organic matter, low fertility, inherent fertility, probably very sandy soils. You need to do everything you can to get nitrogen into those systems. Fertilizer is a really critical, inorganic nitrogen fertilizer is a really critical part of that. But you also need to be encouraging nitrogen retention in those systems and building up that organic matter pool and the microbial community. Otherwise, it's just going to be lost from the system. At the same time, there are systems where that nitrogen uptake process becomes really critical, ones with more inherent fertility, that ability to forage through the soil to maximize AMF mycorrhizal extension, the ability to access microsites and nitrogen as it's remineralizing becomes really important. And so we've sort of taken this experimental work and now built this framework for thinking about where we're at with it and where we want it to be going into the future. Okay, so now I want to sort of get into a little bit, just transition in the last few minutes of our work on biological nitrification inhibition. So I've been intrigued by this plant trait for some time. You can see sort of the work that we've been doing with related to the rhizosphere and plant control over the nitrogen cycle, and BNI is an amazing example of that. And so personally, it's a plant trait based intervention in the nitrogen cycle, which is something we've been excited about pursuing. And it's a potentially high impact approach to improve NUE without synthetic nitrification inhibitors, which have had a mixed bag of success, and growers remain pretty skeptical of them. I work with a lot of conventional growers that really don't like the nitrification inhibitors for a variety of reasons. And this presents an alternative to that. And it's also more broadly a very exciting technology because it's well suited for a wide range of global south cropping systems and climates. And so it can be a way to manage or it can be, one important intervention in managing the nitrogen cycle more holistically in thinking about those plant microbe soil mineral interactions, because that's really where we have to be going with our understanding and management of the nitrogen cycle, is integration and integration of all these different components of the system. And so this leads to, whoops, my last slide, which is work that we are doing with the BNI team, including many people in the room. So, when we first started working on this BNI project, I thought, well, I'd like to really get to know the team and have an opportunity to work with the team on a new project. It's going to take a little while for us to ramp up our experimental work, and also, as I said, I'd had a sort of a personal interest in this plant trait because it was such a compelling example of a lot of the things that we've been working on for some time. And so what I wanted to do was build this team of the CIMMYT BNI collaborators and start to think about BNI in the context of the whole system biogeochemistry. And we looked at the literature, and there really wasn't anything like this out there. There was a lot of things that were informative to developing this much more holistic perspective, but most of them were like, what's the impact on nitrifiers? What is the mechanism of inhibition of nitrification, which I've learned thanks to our BNI colleagues in Aberdeen is very, very varied and complex. And maybe there's been some work on nitrate pools and some work on N2O flux, but there hasn't really been a more kind of synthetic view of what's actually happening in the soil from a biogeochemical perspective when you introduce a trait like this. And so we started out in this work, this synthesis conceptual work. I'm gonna use this. Oh, I was just getting excited to use the button. So we started out over here on nitrifier impacts. And some of this stuff has been more widely studied, if not better known. I'm trying to be very cautious with how I present that because, again, some of the BNI colleagues are like, no, that's really not well known. It's still very complicated and the story is not put together. But I would say that some of this has been more widely studied. What are the direct impacts of these BNI exudates on, for example, the microbes? So we have the ammonia oxidizing bacteria and ammonia oxidizing archaea and Comamox and their enzyme systems. And one of the things I've learned in this process is that there's a lot of things happening. We see a decline in these nitrifier communities. We also see all different kinds of inhibition. For example, you can have those BNI exudates going and gumming up the active site. Sorry. Oh, maybe if I just go back. Great. It's a microbe that I think does co-metabolism of ammonium and nitrate. Amanda, do you remember? So it carries out both ammonification and nitrification. Yeah. And I think that it's a microbe that we're just learning much more about, that it's more widespread than we previously thought. Oh, that might be it, that it carries out both steps in the nitrification. Right. I think that's it. It carries out both steps in the nitrification process. Thank you. So anyway, it can get into the enzyme and gum it up. It can also, these exudates can also like sit in the inner or outer cell wall and create all sorts of problems and disrupt the cell wall, and it can clog up the channels that nutrients move through, and it seems to have some direct inhibitory effect on the microbes themselves, even separate from this, like almost allelopathic. And so we're just sort of learning that there's all these different mechanisms by which these things work. And there's a fair amount of evidence for this impact on the nitrifiers, whatever the mechanism is. This BNI trait does what it's supposed to do. It impacts nitrification. You tend to see an accumulation of ammonium. Okay, so that's where we then said, okay, here's this system where we're accumulating ammonium. And by disrupting nitrate formation, we're immediately going to be limiting or reducing N2O production, this important greenhouse gas, because that's formed from nitrate. We're also going to be limiting nitrate leaching. So again, we need much more, but we have evidence of these disruptions to N2O production and nitrate leaching as a result of blocking this ammonium conversion to nitrate. So then I got to thinking about the fact that microbes see ammonium and nitrate very differently. Microbes have a very strong preference for ammonium, and thus we might expect that there's going to be a lot more immobilization of nitrogen in the soil. So you can think back to that earlier work that I presented of that role of immobilization and taking nitrogen out of that inorganic pool vulnerable to loss, putting it into pools that are more stable and can be turned over when needed. And so we might also expect shifts in the mycorrhizal fungi and overall microbes and saprotrophs as a result of this drastically increased ammonium with respect to nitrate concentration, but also the declines in this community are likely to have feedbacks to this overall microbial community. Also, we know that the exudates themselves are quite tasty to some microbes, not the nitrifiers, but the other ones. There's preliminary evidence that seems to suggest they quite like some of those exudates. And so all of this could result in more microbial biomass, a more fungal dominated microbial community, higher carbon use efficiencies and nitrogen use efficiencies, or just changes in microbial traits. We do have evidence from a biophysicist on the project at Aberdeen that we can actually see more extracellular polysaccharide and biofilm formation. There is some preliminary evidence of more aggregate formation under these crops. And so then you get into this situation where much more organic matter is forming, much more nitrogen-rich organic matter and cycling of nitrogen through the microbial community. And so what we've been trying to do on this project is really scale up this impact on the nitrifiers of BNI to really thinking about the impacts on a whole soil biogeochemical level, because it's our view that it's there where we need to be understanding the nitrogen cycle in order to make gains in NUE. We can focus on one small process, but there is no silver bullet to solving the NUE process. It's a very complicated biogeochemical system. And so when we look at an intervention like BNI, we have to move beyond just thinking about the nitrifier impacts or the short-term nitrate or N2O losses, and really be thinking longer term about the broader biogeochemical changes in that system and how those are going to feed back to the nitrogen cycle. So that has led to us again to go back and visit that earlier framework of nitrogen accumulation, recycling, and loss, and really think about different cropping systems where we might be deploying BNI and where we might be wanting to focus on accumulation, recycling, or loss. And this is part of a paper this is the same paper that we're working on with the team, and really thinking about what are the complementary management practices across this scale of vulnerability that will encourage accumulation, recycling, and uptake. And I don't want to go into that in detail. We're 45 minutes into the talk. It's a little bit detailed. If anyone wants to talk about this, that's fine. But again, this is an effort to sort of integrate that earlier work on the partitioning of N and the biogeochemical processes with what we understand about BNI, and really start to think about and put forward predictions about under what sorts of systems different kinds of complementary management practices to BNI are going to be important and impactful. So I'd just like to say that, this work is done by lots of other people, and Amanda Daly is here. These other people have made contributions, but it's impossible for me to put all of them up on one slide. There's lots of so many postdocs and graduate students and colleagues who have all contributed to this work. And, for that, I'm really grateful. And it's all of these early career people and my colleagues. Not only are they doing this work, but they keep me incredibly excited about moving forward with it. And so I just want to acknowledge that this is a truly team effort to have gotten to where we are today with it. And I'll just end and leave a pretty picture up there from this recent paper, and open it up to questions. Yeah. Thanks.
Anyone has a question here?
Thank you. Can you make sure that the slide, can you move to the slide, 3 slides up with the framework, how BNI?
You want me to move?
Yeah, a little bit up.
I think I can do that.
Oh, okay. Right there, I guess. Okay. Yeah, thank you, Stuart, for this inspirational talk on how to harness microbial community to enhance to understand the nitrogen and carbon cycling or nutrient cycling in general across the agroecosystem, and putting BNI in that context. And there are about 35 online participants, I guess, right? Is that 35, Isabel? 55, sorry.
Wow. Hi, everybody. Thank you for coming. That's
if you
exciting.
have questions, please put that over the chat, and then Isabel will make sure that your question would be asked. Just for the context to everybody, Stuart and his team are also part of the BNI crop sustain in general. So it's how do we quantify the global impact of biological nitrification BNI wheat in enabling the nitrogen reduction or improving the nitrogen use efficiency and reducing environmental impact. And within that, there are 1 to 7 different tracks. Carolina is here, so she's the overall coordinator. But Stuart and many of us here in this room, we specifically work on track 3, which is kind of quantifying the environmental consequences or environmental benefit of BNI by quantifying the nitrous oxide emission potential, nitrate leaching potential, ammonia volatilization reduction potential, or there would be a kind of trade-off on that or something like that, and understanding these processes. So if there are any questions, please please ask. But just to kick off the.
We have already here.
Oh, there is already a question. So can you read that question on the screen here, or?
Oh, no.
Oh, it's
Okay.
not.
No. I
So
can't
here
see
is
them.
the first one. There. It's an excellent talk. Thank you very much. 2 requests. I understand that we should aim high fungi to bacteria ratio for better nutrient use efficiency. I would like to hear your view on agroforestry, syntropic for food forest systems promoted mimic nature biochemical systems. And the second one is, does pruning or grassing or mowing changes microbial activity and composition, with potential effect on types of SLM and therefore nitrogen use efficiency?
Great. Thank you for the question. I think agroforestry can be really well adapted to certain areas in particular, and that is the key, that that it is we're not imposing, some kind of system where it doesn't belong. And so I think where it is where it does belong, one area where they're using agroforestry to quite a lot of success is the northeastern U.S. It's an area that we have had trouble, for example, growing large-scale cropping systems. Trees grow quite well there, and so we've had a lot of interest in integrating, animals with timber production and crops and fruit and things like that. And so if it's well adapted to the region, that's great. And then there was another component of that question. Do you remember? Yeah, so we don't really know the answer to that. Of course, there's been a lot of speculation about fungal bacterial ratios and what they mean. Fungi and bacteria represent a lot of different kinds of microbes, and so we have to be a little careful. We have to be a little careful about generalizations. There seem to be a number of studies, including the work in artificial systems and in the field that Cynthia Kallenbach did, that seem to suggest organic matter formation was associated with a higher fungal bacterial ratio, and yet there's a lot of other data suggesting that that's an oversimplification. So I think we're still figuring out sort of what optimal microbial communities look like in different places. Yeah, I mean, I think, again, mowing is often used to sort of mimic a grazing type of process, and it can improve plant productivity, build organic matter, and again, it needs to be sort of context dependent, and in some locations, I think it can be quite successful.
Okay, thank you, Stuart. The reason that I wanted to keep this on the screen is, the framework of putting BNI into, whether there would be a trade-off. So based on your studies so far, beside the nitrifier communities, are there? I mean, you have stipulated in your framework that there would be consequences on other microbial communities, but are there any trade-off, like having a negative impact on the beneficial microorganisms, so that there would be there could be benefit of putting BNI into the agroecosystem, but there are also trade-off on that?
Yeah, I mean, that's a neat question, and I think that's where ultimately we're going to have to assess broadly the exudation patterns in the soil, because those are the means of communication, the signaling that occurs between the plants and the microbes. And a lot of beneficial microbes are responsive to that kind of signaling process, which really depends on the metabolites, the kinds of compounds that are produced by the plant. And so, I think that that is something that hopefully we're going to learn more about in this project, is what those broader impacts are on the microbes, including those microbes that we might consider beneficial microbes.
And there is one question more towards the beer, but you could also come in, and also, Flavio or Carolina, you can chime in. So is BNI effective in all crops? And what kind of, plant and root physiology would, would have impact on BNI, and how we can harness that in the agroecosystems? There are a lot of literature around where the BNI is effective, not only on wheat, but also on other crops. Carolina, if you want to come in, or if you...
Yeah, we are just starting these studies in different crops, so it's difficult to anticipate if it will work for all the crops. But we are doing some research, in particular in wheat, and the genetics may change based on crops or the different sources that in where we can increase the expression of the BNI activity. And also will depend on the interactions with the environment and how the management of the crop that in particular would be. So yeah, it's several factors that will interact this response.
Yeah, I will follow with a question, Professor Grandy. Based on what you said, I was wondering if in intensive rotation agriculture systems, including a BNI crop, let's say every year, but you have another crops rotating that are non-BNI, would that BNI crop generate any benefit in terms of nitrogen use efficiency in the following crops? Or should we expect through the years a gradual increase in nitrogen use efficiency such that we could lower the nitrogen rate applied?
I think that's a outstanding question. And thinking about the temporal dynamics of BNI and the integration with cropping systems is something that is going on here. And I suspect that there are people in this room that are thinking a lot about that very question. My understanding from the very limited literature out there is that there does seem to be some legacy of BNI, some carryover, but how it plays out over time and what sort of crops are more likely to leave a longer or shorter legacy, and what kind of soil types. I think those are all really important, outstanding agronomic questions. Once we have establish the potential for this trait, I think that it's going to really open the door to exploring it across an incredible range of different conditions. So I think that's hopefully where this all heads next. But again, there's some work in that area. I know there's ongoing work in that area. I know the BNI project has components that are focused in that area. As we build up an understanding of the mechanisms, the processes, the potential, I would imagine that that kind of work is going to probably really become quite dominant in the future, the more agronomy aspects of it, and figuring out where, when, how do we manage this.
Thank you. We're close to hour, but given the interest, we'll still keep for a couple of minutes over here. So yeah, Flavio, just to follow up on what Stuart just said. So there had been literature, and not on a BNI wheat, but in the cropping system where Bracharia is included into the cropping system. The effect of BNI has been also reported into the subsequent crop up to a year or even up to 2 years to some extent. So there are literature, but I think it is opportunity for us to put BNI into the cropping system and how that would have beneficial effect on the temporal scale as well. Are there other questions? Okay, so one last question, Stuart. So your Arun framework, accumulation, recycling, retention, and uptake of nitrogen. So to put the context of BNI into, the context of agronomy, if you had to prioritize one agronomic practice, whether it is a cover crop or organic amendment or reduced tillage or anything like that, that would have a synergistic effect with BNI. I know that many of them would have. What would you choose, and then what would and why it would be?
Can I open that question up to people in the room that are really thinking deeply about the agronomy of BNI? But I will, let's see. If I mean, I don't want to evade the question. So let me just give me a second to think about it. I think that it will be especially ultimately beneficial in systems that have sort of intermediate levels of organic matter. I think that in the short term, it's going to be quite easy to see the impacts of BNI in soils that are really vulnerable to nitrogen loss, and sandier soils where you're applying ammonium to a crop with the BNI trait, you'd expect to see an impact, and we have seen that impact. Those data exist, but I tend to suspect longer term that the real impact of BNI might be through these more indirect biogeochemical mechanisms and sort of the rewiring of the underground system. And that's going to be much more likely in soils that don't fall into that most vulnerable category, but into more of that intermediate category where BNI is helping to support nitrogen retention and recycling. And so I think anything that we can do to support organic matter accumulation in an active microbial community, depending on the type of cropping system, that might be cover crops, it might be using compost or manures or other kinds of amendments, it could be changing tillage, using irrigation. Those kinds of things that are interventions to help support organic matter accumulation, I think are going to really have very positive feedbacks to this BNI trait that maybe are not quite as apparent as the obvious effect of it on ammonium conversion to nitrate in a sandy soil. Right, exactly. So residue management would be a great example of that. Again, we saw that in our earlier work that those soils with more organic matter are mobilizing more fertilizer and moving more nitrogen through the mayom pool, and also producing more available mayom that's accessible to the plants. And so I think that that dynamic will really benefit and create a synergy with that BNI trait.
Thank you. I think with this, let us give Stuart a big round of applause for this stimulating talk. So yeah, a lot of process understanding is kind of an ongoing process, and we would be working together to understand that process together holistically. And we really appreciate Stuart and his team working with us. And then it would be many of the things would be learning curve for us. Maybe some of the applied science that we are doing in the global south would be learning things from for the university and your students as well. So with that, I think we will end it. Before that, I would like to invite our Global Wheat Program Director, Flavio basically to give a minute word of thanks to Stuart.
Thank you for the opportunity, Tech. So I already welcomed, but now I want to thank again the visit of such a distinguished professor and a member of his team here that we see it's a very dynamic team. Very nice drawings in your papers. I found that really nice. So yeah, good example. And we see that we are really engaged and you put your heart in your work. So this is the kind of partner that we cherish very much. Thank you for visiting CIMMYT.
Okay, great. Thank you, everybody.